Welcome back, everyone. So we're in Unit 9. So in this unit, we're going to get into evolution and its process. So this is Chapter 10 in our OpenStax textbook. If you're using Campbell Biology, I believe it's Chapter 13. I can't remember though. So let's get into the unit. So in Chapter 10, we're going to get into, again, evolution and its process. So it's divided into 4 sections. So this video will cover 10.1, discovering how population changes. So by the end of this video, you should be able to explain how Darwin's theory of evolution differed from the current view at the time, as well as describe how the present-day theory of evolution was developed, and then be able to describe how population genetics is used to study the evolution of populations. Okay. So let's start out with some of the history behind the development of this theory of evolution by natural selection. So when we talk about the theory of evolution by natural selection, that refers to basically the mechanisms that that we see in species changing over time. Okay, those small, minute changes that we see over time. Okay, so we're not talking about, big dramatic changes. We're just seeing, how species evolve slowly over time. And this theory of evolution by means of natural selection, it came about through a lot of different ideas that developed over many, many years. So way back in, Plato's time, Plato's view was that species were static and unchanging. But even during that time, there were some ideas of evolutionary viewpoints back then. So it wasn't all, they still viewed that, there was perhaps species that may have existed in the past, or at least that were not present currently. In the 18th century, there were ideas, including, friends of like Darwin's father and things like that, or grandfather, excuse me, that accepted that there were extinct species, so species that no longer existed, that we have fossils of, and they accepted the idea that those species existed in the past and they were no longer there. So that kind of gave the idea that perhaps there were species, that species perhaps were changing over time. There were some famous geologists that understood that, including James Hutton, who proposed that geologic change occurred gradually over long periods of time, that the earth was changing, and it changed gradually, it changed slowly over time, that the earth was not static. In the 19th century, Charles Lyell, who was a friend of Darwin's, argued that the earth was a lot older than previously thought. A lot of people at that time believed in what what the Bible said, literally. And so, when Charles Lyell looked at, the rocks and the fossils and, the striations in the earth, he proposed that the earth was a whole lot older than what was previously thought. And so if the earth was, has changed gradually over a long, long period of time, perhaps so has the species that on the earth. Okay, so, you can see already that, the idea that, the earth is a lot older than was previously thought, and that it's been changing over this long, long period of time. So this started, the naturalist side of things, the biologist side of things, thinking that, maybe, this also applies to organisms as well. So Jean-Baptiste Lamarck proposed that the mechanism for evolutionary change which he referred to as inheritance of acquired characteristics. So he was the first to basically state that traits were acquired by individuals through use or disuse, and it could be passed on to the offspring. Well, this was, proven incorrect. So you can't, suddenly start, using something, and then since you use it a lot, that means that you're going to pass that trait on to your offspring. But he was the first to kind of start thinking that we acquire our characteristics and can pass those on to our offspring. Okay. So there's, a lot of other people that were involved with the theory of evolution by natural selection. So it's not just Charles Darwin. Okay. And then there's two people that were really involved with this theory. Charles Darwin, of course, and then another person, Alfred Russel Wallace. So Charles Darwin, he in 1831 to 1836, he took a trip around the world on the HMS Beagle, and along his way he made stops and he collected a lot of different organisms and studied different plant life and organisms, made lots of observations and took really good notes along the way. In 1848 to 1852, Alfred Russel Wallace, he also, went around the world. One of his stops on his first trip, he stopped in the Amazon rainforest and he collected insects. And then he made another trip from 1854 to 1862, where he spent a lot of time in the Malay archipelago, where he studied all sorts of different organisms. And so, even though these two scientists, Charles Darwin technically wasn't a scientist. He studied theology. He was going to become a priest or a minister, but he decided not to. But these two scientists independently researching. Charles Darwin, he observed on one of his stops, he liked, particularly the Galapagos Islands was one of his favorite destinations that he spent a lot of time on. And he observed that species on these different islands were very similar, but had very distinct differences. And if anybody knows anything about the Galapagos Islands, it's this little cluster of islands of Ecuador. And so it's pretty far off of Ecuador, so it's not like it's easy to get to from the mainland. But it's this little cluster of islands, and the finches there, you can see here some pictures of them, they all look very similar. But they all have very distinct differences. So, he made those observations. Both both Wallace and Darwin, they, were very independent in their observations and their studies. But they were observing similar patterns. So they're coming up with the same hypotheses. Charles Darwin, he called this mechanism where we see like the similarities but with distinct differences, he started thinking that maybe there's this mechanism called natural selection at work. Okay. So what is natural selection? So his idea behind natural selection is based on 3 observations. So the first observation is that the characteristics of organisms are inherited. Okay, so they're passed on from their parents. Okay, so you acquire your characteristics or your traits from your parents. Another observation that he made is that more offspring are produced than are able to survive. So we have an overabundance of offspring. And because we have the fact that we have offspring that vary amongst each other, and that there's variation amongst the offspring that's inherited, there's going to be competition. Okay, so we have this overabundance of offspring. There's variation amongst the offspring. They're going to be competing with each other for limited resources. And so, therefore, the ones that have the best inherited characteristics that allow them to compete best for those limited resources will survive and have more offspring than individuals with variations that are less able to compete. Okay, so it's this idea of survival of the fittest. Fittest means leaving behind the most offspring. And so he termed this descent with modification. Okay. And so he actually collaborated with Wallace. At this time, around this time of 1958, Darwin and Wallace, Wallace was actually really sick. He had contracted something from his trips, and he was really, really sick. So Wallace kind of wrote down all of his ideas, and Darwin and took his ideas and presented his ideas. Wallace, they presented them together, the ideas, before the Linnaean Society in London in 1958. And then finally in 1959, Charles Darwin published on the origin of species, and included on the origin of species, at the beginning of it, you'll see the foreword is posted by Alfred Wallace. So, they were kind of collaborating, but Wallace was very sick at the time, so he wasn't able to publish a book himself, or, really collaborate with Charles Darwin on making the book together. But that's about the history of it. So we're going to watch an Amoeba Sisters video that goes into more of natural selection to give you kind of more of the details behind what natural selection is. And this is a good look into natural selection, because we're going to be working through this idea and the theory behind it throughout the chapter and subsequent videos, or subsequent lecture videos, I should say.
So here's a question for you. Have you ever had strep throat? Now, not all sore throats are strep, but strep throat is caused by bacteria. And if you have had it before, it typically really hurts. And chances are, if you have had it, you've taken a course of antibiotics. Antibiotics are specifically designed to terminate prokaryote cells, which in your body would be bacteria, instead of your own cells, which are eukaryote cells. We have another video all about bacteria and antibiotics, but this video is going to mention a concern we all have right now about antibiotic resistance that can happen in bacteria. It's also the perfect opportunity to illustrate a process that usually takes a very long time to see: natural selection. Now let's explain a basic example of natural selection. Consider these frogs sitting on logs in their habitat. Now let's assume that these are the same species of frogs, so they can breed with each other and they pass down their DNA to their offspring. And naturally, there is variety in these frogs. Some of these frogs are darker green, maybe almost a brown, and some of them are lighter green. There is a variety of traits even in the same species, just like in people. So back to the log. You know what else is in this habitat? Predators. And the predators are finding here that the lighter frogs are much easier to see in this habitat than the darker frogs. So in this particular environment, the darker frogs are having an easier time surviving and potentially more fitness if they can breed. Fitness, in the biological sense, is determined by not how long they live, but by how many offspring they have. These darker frogs pass down their DNA to their offspring so that the new baby frogs will have the DNA from their parents. And the lighter frogs are being selected against, since they are easier to see in this habitat. Now, over a long period of time, you can expect to see a higher frequency of darker frogs. If it continues for a very long time, it could even result in all the frogs in this area being darker. Evolution, which is change over time, could take place because natural selection has occurred. Natural selection is a mechanism of evolution. And one thing to point out, it doesn't necessarily mean that the allele for lighter color is gone completely. The allele could be recessive and carried within the population. And because of processes like crossing over and mutations, there are opportunities for variety. But if this habitat and predators do not change, then the darker frogs will continue to have more fitness. Now, we want to point out that variations and mutations are not things that an organism can will itself to have. Frogs can't just think, it's better for me to be darker, so I'm just going to have some variation to be darker. Because if that was true, well, let's just say I might be a bit different. Mutations and variations, they are random. It's possible they might not have any effect on an organism's fitness. So in that case, the genes are to be passed down if the organism happens to be able to reproduce. Or variations and mutations can be negative. If they are negative and negatively affect the organism's fitness, meaning no babies, well then that trait will not be passed down. But if they have a positive effect on the fitness, then that frog may have more babies than average because that trait is helping them survive and reproduce. It's an adaptation, and over time there will be more babies with those genes, and over time that trait that is an advantage will be more frequent in the population. So what does this have to do with natural selection in bacteria that occurs from antibiotics? Well, keep in mind, there is variation in bacteria as well. For example, some of them might have an enhanced cell wall that makes it hard for an antibiotic to enter, or they could contain enzymes that could assist in breaking down an antibiotic. Now remember, the bacteria didn't will themselves to have those traits. Like our frog example, they are random variations. However, when you take antibiotics, the environment is being altered. The bacteria that have traits that allow them to survive that particular antibiotic will have higher fitness and can reproduce. Bacteria that do not have traits to help them with the antibiotics do not have very much fitness because they're dying, and they can't have fitness if they're dead. Selection has taken place, and the resistant bacteria are the survivors. And once all their competition is killed off, these survivors can reproduce easily with few competition, and every cell that comes from them will inherit that gene that allows them to be resistant to that specific type of antibiotic. Now, bacteria also have this amazing ability to transfer genes to other bacteria, so they could potentially share that resistant gene with other bacteria. It is possible for this specific type of antibiotic to maybe not be as effective for this person in the future. Hospitals especially have this challenge because they are treating multiple patients with bacterial infections in a closed environment. So healthcare workers must have good hygiene practices so that they do not spread resistant bacterial infections from one infected person to another. So this example of natural selection was observable in a shorter amount of time, which can result in evolution in bacteria. But what can be done about this resistance issue? Well, scientists are often in the process of developing new antibiotics. We do have many types of antibiotics available, so scientists hope to stay one step ahead of bacteria. But there are still bacterial strains out there that have developed a resistance to commonly used antibiotics, making them very difficult to treat. So it's important to only take antibiotics for bacterial infections. Antibiotics don't work against viruses, and you don't want to be introducing antibiotics in your body when you don't need them. That will just further select for bacteria that have some form of resistance. It's also important to mention that many vaccines, like the DTaP vaccine, can protect you from serious, potentially deadly bacterial infections such as diphtheria, tetanus, and pertussis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a great kind of introduction into how natural selection works, and we see how natural selection is the mechanism that drives evolution. And so we're going to take a look at an example. So the Amoeba Sisters video gave you the example of antibiotic resistance and how we can see how the antibiotic resistant bacteria was able to evolve just through the population, within the population having a mutation that had some resistance to the antibiotic, and then the bacterial population dying off with exposure to the antibiotic, and then the ones that surviving had that natural resistance to the antibiotic, and then those surviving started to divide, and now we have a population that is resistant to the drug. So that is an example of natural selection within a population. Okay, so here is another example. This example is from your textbook. So this is a study that was conducted in 1976, I believe. So, yep, so we have Daphne birds. So in 1976, basically, they took a look at the beak depth of these Daphne birds. So they basically just measured the beak depth, and they just calculated them all within the population, total of 751 different birds of the same species, so it's the same population. And then you can see how they graphed it. Okay, so you can see that the median beak depth is right around 9.5 millimeters. Okay, so right around there. Okay. But then we see that there was a drought, okay, in 1977. So that reduced the number of small seeds available to the finches. So it caused a lot of the smaller beaked finches to die off because simply there wasn't enough of the food source for them, which they fed on the smaller seeds. So they ended up dying off, okay? And so they came back in 1978 and performed the survey again. And so this time they only had 90 birds that they were able to find. And they did the same thing where they calculated or measured the beak depth of those birds and graphed them. And you can see that the average beak depth shifted. So now it's over 10 millimeters. So it went from an average of about 9.5 to a little bit over 10. Okay, so the population shifted. And you can even see that, if I was to draw a curve here, a little bell-shaped curve, it's kind of over here, but now it's shifted to the right a bit. Okay. So this is an example of natural selection. In this case, it was just an environmental factor, right? So we have a drought that took place, and the ones that had the smaller beaks, they ended up, being the losers in this case. They didn't have enough food. So they ended up dying off, and the ones that were the winners were the ones that had the larger beaks, so they could go and had able to find the better food sources. So they outcompeted the smaller billed birds, and so they were the ones that were able to get enough resources, survive, and pass on the bigger billed trait to the next generation. So we see that the curve has shifted to the right. Okay. So we see that the population was able to, shift it a little, okay? So this is an example of natural selection, okay? All right, so let's talk about variation and adaptation. So natural selection can only occur if there is variation within a population. And by the way, that variation has to be genetic, okay? So you can't deal with variation that is doesn't have anything to do with genetics. So for example, if you get a tattoo, obviously that tattoo is not going to be passed on to your child, okay? So it has to be genetic. So natural selection can only occur if there's variation within the population. So we saw within the last example that there's variation in the bill depth or the bill size of that particular bird species. And then where does that genetic diversity come from? Well, there's two main sources. So we have sexual reproduction. So with sexual reproduction, remember there's two ways that we can get variation with sexual reproduction. So we can get it through the segregation, or the independent assortment of alleles during middle metaphase. Okay, so that was one way. So that shuffles up the chromosomes and how they get distributed into the gametes. The other way was during crossing over. So remember, with crossing over effect, that occurs during which stage? That occurs during prophase part 1. So that creates that recombinant DNA. And so with the exchange of the chromosomes, we can get a little bit more shuffling of the DNA. So that creates genetically unique gametes. Okay, so that occurs through with the shuffling of the chromosomes and everything. We get genetically unique DNA through sexual reproduction. Another thing with sexual reproduction is random mating, with the random fertilization. So that's the third way that we can get some genetic diversity with sexual reproduction. Okay. The other source of genetic diversity is through mutation. So remember, we talked about mutations in last week, and mutations are just changes in the DNA. And so, we talked about all of these different types of mutations. So we can just have like a, base substitution where it just changes switches out one for another. We can have an insertion where that causes a frame shift mutation and so forth. So that can cause a change in the protein that's produced. Okay. So these mutations, there's 3 outcomes to these mutations. One is that this mutation could affect the phenotype of the organism in a way that reduces its fitness. Okay, so it could lower the likelihood of survival, resulting in fewer offspring. Okay, so for example, in that frog example, perhaps the mutation causes the frog to produce a color that makes it stand out on the log for the bird. Okay, so the bird is now easy, can find the frog easier, and the frog is going to be lowers the likelihood of its survival, so it's going to get eaten before it reproduces. So that would be considered, a negative mutation, right? A bad mutation. So it's going to lower its fitness. Another possibility is that the mutation may produce a phenotype with a beneficial effect on fitness. So maybe it produces a protein that causes the pigmentation of the frog to blend into its surroundings better. Okay, in doing so, maybe that would be a benefit, so that the frog it can't be seen by the bird, and if the bird can't see the frog, then the frog won't get eaten because it's hidden in plain sight. And so that would be a beneficial mutation. Okay, that increases the fitness. Okay, or maybe the mutation has no effect on the fitness. Okay, so maybe the frog mutation just causes maybe an extra little bump to form on its skin. Okay, it doesn't really affect the frog in any way. Okay, has no effect on it. It doesn't change its fitness. It doesn't affect its survival. No effect whatsoever. So that would be a neutral mutation. Okay, so It doesn't do anything. Okay, so mutations could be detrimental to its survival, it could be beneficial to its survival, or it could have no effect. Okay, so it just kind of depends on what happens. Remember, mutations are random. Okay, so it's not like the frog, decides, okay, I really want to change colors today. I really hope I get a beneficial mutation, right? So it's completely random what they end up getting. Okay. And so let's get into patterns of evolution. So we know that natural selection is the mechanism that drives evolution. So what is evolution? So evolution of species has resulted from enormous variation in form and function. So it's just all of those, natural selection just is, all the accumulation of those adaptations. Now, we do have several different types of evolution. So divergent evolution is evolution resulting in different forms in two species with a common ancestor. Okay, so that means that we have two species that came from the same common ancestor, but they basically evolved over time into different forms, okay, based on whatever the situation was. So for example, if we have like two flower species, one that gets pollinated by a moth at night, so it only opens at night, versus the moth or butterfly that pollinates it during the day, it only opens during the day. It might evolve two different colorations or something like that. So that's divergent. Came from the common ancestor, but this one has a mutation that produces a coloration and evolution strategy that its pollinates it at night versus during the day. So it evolved over time to form two different species. So that's divergent. So coming from one species splitting into two species. So they're diverging from each other, so they're becoming two different forms. Convergent evolution is the opposite. So you're basically taking two completely different species, but if you look at them, they look very similar to each other. Okay, so they're completely unrelated, but they look very similar to each other. Okay, so they evolved similar traits, but they're actually not related. Okay, so for example, if you look at a shark and a whale, okay, so sharks and whales, they have completely different evolutionary histories, but they look very similar. They have, basically they're swimming in the ocean, but one has gills and stays underwater all the time. The other one, breathes air. So different breathing strategies. One's a mammal, one's a fish, but they look very similar to each other. So similar forms, but completely different species. So they evolved to live in the same environment, but they don't share a common ancestor. Okay? So divergent evolution, they share a common ancestor, but they evolved to live in different environments. Convergent evolution, they don't share a common ancestor, but they evolved to live in the same environment. Okay, so they have similar forms. All right. So moving on. Okay, so we learned some of the terminology, and we'll be using more of that terminology and giving more examples in the next couple lecture videos. So let's get into modern synthesis. So when we're talking about Darwin and Wallace and their development of the theory of natural selection or theory of evolution by means of natural selection, when they developed that idea of natural selection, they were completely unaware of what Gregor Mendel was doing with his garden peas over in what is now Austria. Okay, so even though, Gregor Mendel, I mean, he published his stuff back in 1866, and Charles Darwin published his book in 1859. So, you know, all this was happening right around the same time. But they had no idea what each of, you know, Mendel and Darwin, they don't know that they were doing these things at the same time. Okay, so they weren't in communication with each other. And because of that, if we take a step back, you know, now looking at today, we can see that Darwin and Wallace's idea of natural selection and Gregor Mendel's work in genetics, they overlap. Okay, so but that overlap, that connection wasn't made until about the 1940s. So modern synthesis is what it's called. It's this overarching evolutionary paradigm that took shape by the 1940s, and that's generally accepted today. So that describes, so what the modern synthesis means is that it describes how evolutionary pressures, such as natural selection, affect a population's genetic makeup. Okay, so in turn, how this can result in the gradual evolution of population and species. So basically, if we take a look at the alleles, okay, so remember, if we're looking at, you know, going back to Gregor Mendel and looking at his garden peas and looking at, you know, the percentage that are heterozygotes versus homozygous dominants versus homozygous recessives within a population. We call that microevolution, by the way. So the changes in a population's genetic structure or allele frequency, okay, we can apply that to natural selection, okay? So if we see changes within the allele frequency, the percentage of the population that has the dominant allele versus the recessive allele, if we see changes in that over time, that's evolution. Evolution is just change in genetics. It's a genetic change over time, okay? So if we see those little changes, that's called microevolution. That based on natural selection, okay? So if, you know, population, certain traits are more better adapted to a particular environment, they survive longer, they get passed on to the next generation. We'll see the allele frequencies change. The processes that gave rise to the new species and higher taxonomic groups, those changes over a long period of time, they accumulate, become more and more widely divergent, becomes macroevolution. So macroevolution is a broader scale of evolutionary changes seen over a long period of time, okay? So this means long period of time, okay? So microevolution is just small changes in allele frequency within a population, you know, generation to generation or, you know, year to year. But if we take thousands of generations, add up those changes, we're going to get new species. Okay, so that's at the macroevolution level. So microevolution level is at the population level, just small changes over time. Macroevolution level is speciation. Okay, these changes get to the point where we have the populations have diverged to become so different that they can no longer interbreed together, so they're no longer the same species. So we've become new species. Okay, and we'll talk more about speciation, I think, in the third lecture. All right, so this is this idea of modern synthesis, where we're bringing together Darwin's ideas and Gregor Mendel's ideas, and we're putting it all together. Okay, and that brings us to population genetics. So population genetics, we've actually already talked about a little bit already with Hardy-Weinberg. Remember that equilibrium? And we'll talk more about that. Okay, so population genetics is where we track allele frequencies in a population over time for a given trait. So for example, we can track the frequency of the black allele versus the pepper color allele in a population of moths throughout the Industrial Revolution. Okay, so you can see here we have two alleles, the dark color allele or the black allele, and then the allele that causes the pepper color. And then you can see in the original population, the frequency of the pepper allele was a lot more common. Okay, so that allele was more frequent. But then with the Industrial Revolution in England, it pumped out all the soot that was pumped out of the smokestacks, covered all of the trees with the soot. And so it killed all the lichen that was growing on the trees. And in doing so, basically it made the pepper moth, the pepper colored moths stand out to its predators. And so the dark colored moth, they were more camouflaged against the sooty colored ash that was covering all the trees. And so they started to survive more and had the better adaptation. And so now we see the population shift towards the darker color moth. So we see the allele frequencies change. Okay. So we're going to watch a video on Hardy-Weinberg. So it will be a little bit of review since you already played around with the equilibrium, but it's an amoeba scissors video, so it should be fun and helpful as well.
Do you like math? Sometimes when people are starting out in biology, for some reason they assume there is no math in biology, but then they soon find out they were wrong. My math teachers were telling the truth when they said math isn't everything. Chi-squares, osmotic pressure calculations, standard curves with gel electrophoresis, Punnett square ratios, these all have math. But one of my favorite topics that involves biology and math, and is also absolutely fascinating, is the Hardy-Weinberg equilibrium. It got its name from a mathematician and a physician, and it states that a population's allele and genotype frequencies are constant, unless there's some type of evolutionary force acting upon them. A reminder from our ecology videos, a population in this case is a group of organisms that are all the same species and can breed with each other, and have fertile offspring. However, there can be variety among the population as they're not clones of each other, but based on Hardy-Weinberg equilibrium, the population's allele and genotype frequencies would remain constant if no evolutionary force acts upon them. If we have these frogs here, assume they are all the same species in this example, but there are some slight differences. In our simplified example, some of the frogs are lighter green and some are darker green. We're going to use a simple genotype in this example. All frogs in this population will be either big G, big G, big G, or little g. There's also an allele frequency in our population. We're going to say here that 60%, a frequency of 0.6, of the alleles are big G. 40%, or a frequency of 0.4, of the alleles are little g. Notice the percentages add up to 100%, and the frequencies add up to 1. Now, to be in Hardy-Weinberg equilibrium, we have to have 5 assumptions. Number 1, no selection. No natural selection is acting upon these frogs. That means neither dark green nor light green will have any impact on reproductive fitness. Number 2, no mutation. Baby frogs inherit genes from their parents and there are never mutations. Number 3, no migration. Frogs can't come in, frogs can't go out. Number 4, large population. There are a lot of frogs. It turns out small populations are more vulnerable to genetic drift, by the way. See our genetic drift video. Number 5, random mating. The frogs mate without any specific choice. All of these 5 assumptions must be kept in order for Hardy-Weinberg equilibrium to happen. So, in real life, does this generally happen? No. For example, in real life, maybe in a certain environment, predators can more easily see the light green frogs, so perhaps they are eaten more and have less reproductive fitness. So if Hardy-Weinberg equilibrium is unrealistic in nature, why does the Hardy-Weinberg equilibrium matter? That's where the math comes in. The Hardy-Weinberg equilibrium gives you this baseline to compare how an evolving population could compare to one that remains constant without evolutionary forces acting upon it. And now let's explore the math. So there are two equations in Hardy-Weinberg equilibrium that we will focus on. We're going to start with the first one. P plus Q equals 1. In this equation, P is the dominant allele frequency in the population, and Q is the recessive allele frequency in the population. By the way, Hardy-Weinberg equilibrium doesn't mean that P has to equal Q, and the dominant allele frequency in any population doesn't have to be larger than the recessive allele frequency in a population. That's a misconception because dominant alleles aren't always the more common allele. The equation does say that the dominant allele frequency and the recessive allele frequency have to equal 1. So, in my example here, if I say 60% of the alleles are big G, 40% of the alleles are little g, 0.6 is P, and 0.4 is Q. So this equation, P plus Q equals 1, is for those allele frequencies. But what if I wanted to know the genotype frequencies? Meaning, I wanted to know the frequency of frogs that are homozygous dominant, which is big G, heterozygous, which is big G, little g, or homozygous recessive, which is little g. Then I can use this other Hardy-Weinberg equilibrium equation, P squared plus 2PQ plus Q squared is equal to 1. So P squared is the homozygous dominant frequency, the frequency of big G in this case. 2PQ is the heterozygous frequency, so big G frequency in this case. Q squared is the homozygous recessive frequency, so little g frequency in this case. Let's plug those previous P and Q values in. P squared would then be 0.36. 2PQ would be 0.48. Q squared equals 0.16. Cool, huh? The only thing is that when calculating Hardy-Weinberg equilibrium problems, you don't always have the P and Q values. So we're going to do an example. New population of frogs, so forget the previous frequencies. This is a new frog population in a new frog land, but we'll still use the same allele letters. So here's the information you get for this new population. There are 500 frogs, and of those, 375 frogs are dark green. The rest are light green. With that information, please solve all genotype frequencies and all allele frequencies if in Hardy-Weinberg equilibrium. Step 1. Determine first whether you're going to work with the first equation or the second equation. Since I'm working with individuals here that have genotypes, I'm going to work with the second equation first. Step 2. Figure out what value you can determine. So 375 frogs are dark green out of 500 total. That would mean 125 frogs are light green since there are 500 frogs total. But I can't use whole numbers like that, as both of these equations ultimately are equal to 1. I need frequencies for these equations. Now I could say 375 dark green frogs out of 500 total frogs is 0.75, but all I know is that would be the frequency of dark green frogs. The problem is dark green frogs could have genotype big G, or they could have big G. I can't assume they're one genotype or the other, so I shouldn't use that value. The recessive genotype, resulting in a light green trait, is safe to use though, because I know that light green frogs are the genotype little g. They can't be anything else. So 125 frogs out of 500 frogs equals 0.25. That means, from the equation, the value Q squared equals 0.25. Step 3. Take a value you solved from the previous step and calculate from there. So if I know that Q squared is 0.25, then I could go ahead and solve for Q. If Q squared is 0.25, I can determine Q if I take the square root of 0.25. Therefore, Q is equal to 0.5. That's the allele frequency for the recessive allele little g. If I know the Q value, I can find out the P value using the first equation. Since P plus Q is equal to 1, now I know that P is equal to 0.5. That's the allele frequency for the dominant allele, big G. Now I have P, and I have Q, and I can take care of everything else. I can use the second equation to determine the homozygous dominant frequency, the heterozygous frequency, and I already knew the homozygous recessive frequency from the beginning. Plugging those in, P squared equals 0.25 for the genotype frequency, big G, 2pq equals 0.5 for the genotype frequency big G, little g. Q squared equals 0.25 for the genotype frequency little g, little g. Phew, that's a lot. Finally, some last tips for solving Hardy-Weinberg equations. Number 1, I used nice numbers, meaning I didn't need a calculator for my examples. It's not always like that. You might need a calculator for the numbers you're given, and you might need a round. Number 2, when you get your final results, always check that the values you calculated do equal 1 for both equations. This can really help you check your work. Number 3, be careful not to assume too much with given info. Remember when I was given a value of 375 frogs out of 500 being dark green? I didn't necessarily know whether they were big G, or big G, little g. Instead, I chose to work with the recessive genotype since I knew light green frogs would be little g, little g. Number 4, practice. There are a lot of practice problems you can find online, and remember, this skill can be very useful for determining the extent of evolutionary forces because you have this base comparison of Hardy-Weinberg equilibrium to compare it to. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again with the Hardy-Weinberg equilibrium, we can see that basically we can use that to see any shift in the population's allele or genotype frequencies. But again, when we're using this equation, we have to remember that we're just assuming that it's inherently stable. Okay, the population is stable at that snippet of time that we're looking at. So if there's some sort of evolutionary force acting on the population, it's not really going to work. But so, you know, according to the video, as well as your textbook, there are, you know, these different forces that can affect the Hardy-Weinberg equilibrium, which would make it not work. So one being natural selection. So Hardy-Weinberg equilibrium assumes that that every, you know, that natural selection is not a part of it. Mutations, that there's no mutations. Genetic drift, we'll talk about genetic drift, I believe, in the next video. Yeah, so genetic drift is not a part of it. There's no migration into or out of the population, and that there's random mating. So obviously that's not something that occurs normally with populations. So populations usually do non-random mating. Okay. So again, with the Hardy-Weinberg equilibrium, we did have some practice using it, so I'm not going to go into too much detail with it. But again, that's basically, we can compare the allele frequencies using the Hardy-Weinberg equilibrium, and then that way we can confirm if the population has changed over time. So if we look at it in, for example, in the previous, like if we look at it in 1886, and then look at the allele frequencies of the dominant allele versus the recessive allele in 1886, and then compare it in 1896, and we see it change, then we know that evolution has taken place. Okay, so that's how we can figure that out. All right, so we're going to do some review questions. So the first question, which scientific concept did Charles Darwin and Alfred Wallace independently discover? Was it A, mutation, B, natural selection, C, overbreeding, or D, sexual reproduction? If you chose natural selection, I would agree with you. Sexual reproduction, I think people have discovered that a long time before before Charles Darwin. Yes, so natural selection is what they independently discovered. So they both kind of, figured that out on their own through their travels, and then they kind of came together and presented it at the Linnaean Society in London. So B is the correct answer. Here's another question. Which of the following situations will lead to natural selection? Is it A, the seeds of two plants land near each other and one grows larger than the other? B, two types of fish eat the same kind of food and one is better able to gather food than the other? Sorry for the misspelling. C, male lions compete for the right to mate with females with only one possible winner. Or D, all of the above. So which one of the following situations will lead to natural selection? If you chose D, all of the above, I would agree with you. So all of these are basically showing some sort of competition, so survival of the fittest. So this is examples of natural selection. So D, all of the above. All right, so what is the difference between micro and macroevolution? Is it A, microevolution describes the evolution of small organisms such as insects, while macroevolution describes the evolution of large organisms like people and elephants? Or is it B, microevolution describes the evolution of microscopic entities such as molecules and proteins, while macroevolution describes the evolution of whole organisms? Or is it C, microevolution describes the evolution of populations, while macroevolution describes the emergence of new species over long periods of time? Or is it D, microevolution describes the evolution of organisms over their lifetimes, while macroevolution describes the evolution of organisms over multiple generations? So what's the difference between microevolution and macroevolution? If you said C, microevolution describes the evolution of populations, while macroevolution describes the emergence of new species, I would agree with you. Okay, so remember micro, that's at the micro level, so looking at populations, how they change over time. Macro, that's looking at how the accumulation of all of those changes within a population could cause it to change so much that two populations of the same species may become so different that if they were to interbreed, or if they come together again, they no longer can interbreed, so they've become new species through macroevolution. So C is the correct answer. And finally, we have population genetics is the study of, is it A, how allele frequencies in a population change over time? B, population of cells in an individual? C, the rate of population growth? Or D, how genes affect embryological development? What is population genetics? If you said A, how allele frequencies in a population change over time, I would agree with you. Okay, so population genetics, we're studying those allele frequencies. Okay, so the P plus the Q. So if those allele frequencies change over time, that's what we're studying with population genetics. Okay. All right, so we're done with our first section of chapter 10. So in chapter 10.1, discovering how populations change, you should now be able to explain how Darwin's theory of evolution differed from the current view at the time. You should also be able to describe how the present-day theory of evolution was developed, as well as describe how population genetics is used to study the evolution of populations. So for the next video, we're going to get into the mechanisms of evolution in 10.2.